some of these issues, scientists have come up with a somewhat more precise way of doing things, of bringing in new genetic variation. So we call that genetic engineering. And you'll see that the term engineering is maybe used a little bit loosely, especially with this first approach here. So with genetic engineering, what's different compared to the mutagenesis is that we're actually going out into other species and looking for genes that do something that we're interested in, and then moving those genes into the plant that we want to work with. And so based on what we talked about, what Amanda talked about last week, the genetic code, why is it that we would be able to, say, take a gene from an animal and put it into a plant and have it work? Would it work?
I mean, it depends. You have to be careful. I mean, you're talking about splicing here. I mean, like, I'm sure certain ones would. Like, they took the glowing jellyfish and they put it in a fly, and I mean, that worked, but you have to just be careful about what part you're, I mean, that seems kind of obvious, though. You can't just, like, throw some DNA in something and expect it to work, except for in the lab today, because that's what it sounded like. We're just throwing some DNA and expecting it to work. But in general,
so in terms of taking like a DNA sequence, say from an animal, is there any reason that it couldn't be turned into a protein in a plant? So what's the process for doing that? We're starting with DNA. What's the next step to get to a protein? Yeah, make RNA. And then what's the next step? You get a protein, right? So we do transcription, where we make the RNA copy, and then translation, where we go and attach the amino acid that corresponds to each of those 3-letter RNA codes, and that's how we make our protein. And so the genetic code is actually shared by all organisms. So from bacteria to animals to plants, it's all the same. So that means that you can, in theory, take the gene from any organism and move it into any other, and you'll be able to make a protein. Now, whether or not making that protein has negative consequences for the organism you put it in is another story, but you should be able to make a protein. And so the first step to doing this is a process called cloning. And so we talked about actually almost all of the components, all of the steps required for cloning over the last 3 or 4 lectures. And so the first step is identifying the gene that you're interested in. So, let's say we want a disease resistance gene for corn. Turns out rice has that gene. So we want to take the rice gene and then put it into corn. So we find the gene, and then we need to make copies of it. So does anyone remember an approach to make copies of DNA? There's a whole song about it.
PCR.
That's right, so PCR. So say we've got our, this will be our rice genome here. So let's say that our disease resistance gene is right here. So then we'll design primers, which are those short little pieces of DNA that are specific to that gene we're interested in. And they'll kind of line up on either end of the gene. And then we'll do PCR, and then we'll make a whole bunch of double-stranded copies of our gene of interest. So now we've got a tube with a whole bunch of copies of that gene. But it's many copies, but it's not really enough to go through and move it into other species. And PCR is also not very efficient for making copies of very, very big things. And so what we're actually going to do is we're going to hijack the bacterial DNA replication process to make even more copies of this for us. And so you might remember something called a plasmid. So So a plasmid is a circular piece of DNA that are found in bacteria. So bacteria can replicate these at very, very high levels. So this will be our plasmid here. All right. So the next step is to actually put our DNA from the rice into the plasmid. So to do that, we're going to use restriction enzymes. So can anyone tell me what a restriction enzyme does? We used them in lab last week. What's that? Yeah, so they cut DNA and they do it at specific sequences. So what you'll do is you'll find a restriction enzyme that you can use to cut the ends of your PCR product here from the rice. So I'll make a little cut here. And that same enzyme will also need to be able to make a cut on your plasmid. So let's cut our plasmid like that. And then what you'll do is you'll put your PCR product that's been cut in a tube with the plasmid that's been cut, and then you'll add another enzyme to help stitch everything together. And then what you'll wind up with is this circular plasmid in the bacteria that is now making tons and tons of copies of your gene from the rice. Yeah?
Wait, so in biotech engineering, they're using bacteria to grow new, wait, is that right, to grow new bits of DNA coding for anything?
Mm-hmm.
And they're using, wow.
Yep.
Right.
Yeah, so this is a process that I use it quite a bit in my lab. So if there's a gene I'm interested in studying, I'll clone it out of the plant that I work on, and then put it into E. coli, actually, as the bacteria I use, to make more copies of it so I can use it for other experiments. Yeah. All right, so now we've got lots and lots of copies of our rice gene. So now we need to get it into our corn plant.